I thank everybody for logging on today. And for those that aren't here, you can pass the word along that if they want to be added to the email list, that they can feel free to email me. And I'm going to pass this along to Janet to introduce our speaker for today so that we don't waste any time.
Thanks, Renee. Today we have Dr. Mike Humphries with us. Mike is currently the head of U.S. Medical Affairs for AnHeart Therapeutics. He has developed extensive experience in drug discovery, early development, and medical affairs in roles for several companies, including Array Biopharma, Bayer, Ariad, and Takeda. Most recently, Mike was with Takeda for over 7 years, where he developed expertise founded in precision medicine and oncology, focused towards lung cancer. In his former role, he led the lung franchise for global medical affairs by designing the medical strategy, the key opinion leader engagement plan, as well as evidence generation plan that aligned with this strategy, and delivering on medical tactics. He is based in Denver, Colorado, where he lives close to the mountains with his wife and 2 children. He is an avid rock climber, skier, and a general enthusiast of mountain culture and lifestyle. Welcome, Mike.
Thanks, Janet. Very much appreciated. I hope everybody can hear me well. I'm going to kick it off by just introducing my colleagues. We actually have a really good team of AnHeart personnel here today. So I think any question that comes our way, you will have one expert that can answer it for you. So I'm just going to call off a couple of people, and I would ask if you could just introduce yourself pretty quickly. Leanne, let's start with you.
Hi, my name is Leanne. Leanne Lee. I'm the US CMO of AnHeart Therapeutics. Very pleasure to be here. Thank you.
Jim, thanks, Leanne. Jim, you're on mute.
Good morning, everyone. Jim Koska, Vice President of Clinical Operations. Partner closely with Leanne and Mike, and look forward to the discussion today. Thank you.
Thanks, Jim. Heinrich, can you introduce yourself, please?
Yeah, hello, it's Heinrich Farren. I'm the medical director for most of the North American and European medical monitoring for our current study that's going on. I'm really happy to be here, and let's see what you have. It's great. Thank you.
Thanks, Heinrich. Emeka, can you introduce yourself, please?
Sure. Hi, everyone. Emeka Seppo here, Director of Clinical Operations, responsible for driving our global G2A trial. So, looking forward to working with you and collaborating with you.
Thanks, Emeka. And you guys are lucky. Our CEO is actually on the phone today. So, Jerry, if you don't mind introducing yourself.
Yeah, hi everyone. I'm CEO and the co-founder of the company, and I hope that with my colleagues' support, that we can bring this drug to patients as soon as possible.
Thanks, Jerry. And that's it from our side. So, Janet, if you don't mind sharing the slides, I can get moving. Great. Thanks very much. So, we're going to talk about taletrectinib today, but we're also going to talk a little bit more beyond that. And I'm going to try and introduce you as well to kinase biology, as well as some of the, what makes a kinase tick, and then also some of the key clinical trial endpoints that I'm going to go through with respect to this presentation. So we'll probably start off with some of the more introductory material, and then the latter half of this will focus 100% on taletrectinib, a next generation ROS1 tyrosine kinase inhibitor, obviously being investigated in ROS1 positive advanced non-small cell lung cancer. So if you could go to the next slide. There is one disclaimer slide that's provided here. Important to note that I, as well as my colleagues on this call, are all employees of AnHeart Therapeutics. The information provided here is purely educational and informational in nature. This should really not constitute providing any medical advice or professional services within the context of this call. We can't really provide any medical advice today. That obviously should be a discussion between your personal physician and yourself. So with that being said, I'll go ahead to the next slide and get started. So just a little level set before we begin. Janet mentioned some of these things, but I like to make my presentations warm and welcoming. So a little bit about me. I've lived in Denver, Colorado for over 20 years. I'm originally from the East Coast, the small state of Delaware. I moved here for college and I never left the state of Colorado. And that probably has a lot to do with the offerings in the state. As Janet mentioned, I'm a lover of all things mountain culture. I'm a big skier, a big rock climber. I've pushed this on my family as well, perhaps a little too hard sometimes. I have 2 kids, a wife. We live in the suburbs, and I'm a big lover of science as well. So I really focus on my job and on having fun in the mountain environment around here. With respect to science, I've been in drug development greater than 16 years. Almost all of that has been in kinase biology and precision medicine across a lot of disease states, but probably the last 8 to 9 years really in lung cancer is where I've been focusing. My dream one day is to hopefully in retirement be a ski patroller, one of those people that wears a yellow vest and tells people to slow down. So we'll see if I eventually get there or not. Again, this should be a transparent presentation. You are welcome to write questions that you have in the chat. You are welcome to interrupt me as we go, and I will do my best to be as transparent and address any questions that I can answer. Obviously, another quick reminder that I can't provide any medical advice. What I really hope you get from this presentation today is really the following that's shown here. First of all, an understanding of the activity or the efficacy and the safety profile of taletrectinib from our clinical trials. And obviously, the corresponding clinical development program, and most importantly, our pivotal trial, meaning the one that's going to be used for market authorizations and regulatory approval. We'll talk about that. That trial is known as TRUST-II. And then also the potential for this drug, taletrectinib, to address unmet treatment needs in people living with ROS1 positive non-small cell lung cancer. And a couple of comments associated with this statement. I think in all cancer therapy, the goal is always going to be moving forward and gaining cures. I think that's really important to state, and I think that's why we continue to do this and try and develop novel therapies or novel mechanisms of surveilling disease or identifying it early. All of that is going to really roll together to increase cure rates. Importantly, though, I can certainly recognize that each individual on this call is going to probably have a different idea of what an unmet need really is as it relates to their specific case. And I won't go into what those will be, but from a high level, the way we think about this in the ROS1 disease landscape is really addressing long-term disease control with perfect quality of life. I think that's one. I think secondarily, it's being able to address disease that is spread to the brain. And then finally, it's being able to address acquired resistance mutations that emerge on prior therapy, meaning an individual responds to a therapy, but then that drug stops working with a specific amount of time. How do we address that? So we'll talk about some of those things as I move forward. So Janet, if you could go to the next slide. So a little bit of introduction for you and a level set on kinase biology. I've worked in this field for a long time, and I'm going to try and do my best to distill it down for you. So let's go through some of these questions and obviously pictures that I've included as well. So what is ROS1? So ROS1 is a protein tyrosine kinase that exists within cells. Tyrosine, so to step back a little bit, I think you could think of a protein as a beaded necklace, for instance, with each one of those individual beads representing an amino acid. And when that rolls all together into the necklace, that would be the actual protein. So amino acids are really the building blocks of proteins, if you will. Tyrosine is just one of them. There's actually 20 to 22 of them but tyrosine is one of those specific ones. And we'll talk about the action that it does on that in a moment here. As you know, ROS1 is mutated and genetically aberrant in about 1 to 2% of non-small cell lung cancer cases. So what is a protein kinase? So protein tyrosine kinases are enzymes. Enzymes are the doers of the cells. They're the proteins within cells that conduct chemical reactions. And what they do is they add phosphate to target protein tyrosines, meaning that amino acid that's going to exist in that target protein. And we call these quote unquote substrates. So this reaction is called a phosphotransferase reaction, and it's shown in the diagram on the bottom left here, with the K in the Pac-Man type feature representing a kinase, the green that says sub representing the substrate. And what this kinase does, it kind of has a pocket that exists within it. We call that an active site where that chemical reaction, that phosphotransferase reaction occurs. And they take ATP, which is the energy equivalent of the cell. They cleave one phosphate off of that ATP, and they simply add it to that substrate. And that transmits signals through cells. And it's a variety of different signals that could occur. It could be a signal to grow. It could be a signal to stop growing. It could be a signal to move to a different location within the body. It could be a signal to express different proteins within the cell. There are a variety of these things that could happen. This is not dissimilar from what you experience by going to the Starbucks drive-thru, where you stop at the window, you order your coffee, and then your coffee comes out on the other side. It's simply a signaling mechanism that occurs within cells. So if we could go to the next slide. So what happens to ROS1 in cancer? I'm going to step back a little bit here and talk about the ultimate dogma that occurs within biology. And that is that DNA exists as the blueprint, if you will, of information within cells. And that information is really coded within genes. That information is eventually translated into protein. So any information that you see in DNA impacts the structure and the code of that protein. The proteins are the molecules within cells, again, that are the doers. They're the functional units inside a cell. So if you have ROS1 and it undergoes genetic changes, it fuses to another gene, that gets manifest or reflected in the ultimate protein, and you have a fusion protein. These kinases, like ROS1, are highly, highly controlled within normal cells. And in cells that have gene fusions, like ROS1 gene fusions, that normal kinase won't turn off. It's continually transmitting those signals. So you could imagine, with the Starbucks analogy again, that if you order one glass of coffee, it's going to come back with hundreds of them. And that's what really happens. It just continues to signal and does not turn off. So how do we target ROS1 in cancer? You can imagine if it's overactivated that we need to inhibit it some way or another. And we do that with specific drugs. And these drugs are called tyrosine kinase inhibitors. So tyrosine kinase, meaning ROS1 is a tyrosine kinase, and inhibiting that specific molecule with a small chemical substance. The way it does this is it competes with ATP for that active site, and it blocks that phosphotransferase reaction so that phosphate does not get transferred to the substrate. You can see this again in the diagram, same one we saw on the previous slide, where the ROS1 TKI blocks ATP's entry, it blocks the substrate's entry, phosphate does not get added to the substrate. So what might happen in acquired resistance? Well, this again is the case where a drug might work. It might work for a specific amount of time, whatever that amount of time is, and then it stops working. One of the mechanisms that could cause this, and this is by means not the only mechanism, is specific mutations or changes that occur in that kinase so the drug can't bind. So you can see this again described in the diagram below, where the kinase now has this extra piece hanging out into the active site, and that specifically blocks the TKI from binding, and it still allows for that phosphotransferase reaction to occur, and phosphate's going to get added to those downstream substrates, and the signals will continue to transmit even in the presence of that drug. This is reflected in a more biological way in the diagram on the right here, showing crizotinib-resistant mutations that occur. This is not the change in the protein that occurs when crizotinib binds, it's just showing the location of these mutations. And you can see a lot of ribbons and different colors and things like that, but you can imagine there's kind of a Pac-Man looking face facing towards the camera a little bit and a little bit to the left. And you can see the crizotinib molecule actually sitting in there. On the outskirts of where crizotinib is where some of these, what we call solvent front mutations exist. Solvent front meaning that it actually contacts the liquid on the outside of the protein. G2032R is one of these. Deeper in that pocket, we see the gatekeeper mutation, which is L1951R. So these mutations exist in different places within the proteins. They can change its overall fold and its overall structure and influence drug binding. So that is my spiel and minimal lesson on kinase biology. I think I'm going to go to the next slide now if there are not specific questions associated with this. So we're going to talk today about taletrectinib, and we're going to talk about specific clinical trial endpoints. And please don't panic when you see this slide. I realize there's a ton of words, a lot of definitions. This is really provided for your benefit. I'm not going to go through each one of these. I'm going to go into specific ones and talk about those. And on each individual slide, I've actually, if there's a endpoint I'm highlighting, I've actually provided that, that definition on that individual slide so you can review it again. So Janet, if you could go to the next slide. So these are the ones we're really going to talk mostly about, not exclusively, but just mostly today. We're going to talk about something called an ORR, or an objective response rate. You could also call this an overall response rate, and I won't really differentiate those. They largely mean the same thing. But this is really a measure of tumor shrinkage, or tumors disappearing altogether. It's a rate, so it's a percentage. It's a percentage of individuals in the trial whose cancer shrink or disappears. And this could be a complete disappearance. That would be something we call a complete response, or it could be a partial disappearance. And that would be something we call a PR, or a partial response. And you'll see the numbers associated with this, but what this really means is there's shrinkage greater than 30% of the tumors that are being measured. We're going to talk about ORR in the brain. So this would be an intracranial or an IC ORR. So that's specific from the neck up. It's tumor lesions in the brain that have shrunk or gone away. And most importantly, we're going to talk about the safety, or the adverse AE event profile. This summarizes the specific types of adverse events that occur, how severe they may be, and this is measured by the grading, grade 1 through 5. And the number of participants that have been afflicted by that, and that's measured in a percentage. So Janet, if you could go to the next slide from here. We're also going to talk about a couple of specific types of figures. I've highlighted those here. I'm not going to show you any figures beyond these specific diagrams. One of those is a waterfall plot that is shown on the left. And this is really kind of a deeper look at tumor shrinkage of what you'd see really in an ORR that we talked about. So the two red arrows that are shown show the 20% mark increase in tumor size and a 30% decrease. Anything largely that would fall below that 30% line would be a partial response. Anything that would fall above that 20% line, we would call progressive disease. And from left to right, these graphs usually show individuals whose tumors have grown, and then on the far right side, the greatest best response, or greatest tumor shrinkage that an individual has experienced. So each one of these bars represent one individual participant. On the right, you can basically see a spider plot, and this is tracking objective response, ORR, but it does it across time for each individual. And you can understand trends about how much tumor shrinkage occurred and how long that lasted by looking at a spider plot. So if you look at the kind of light pinkish traces that you can see on the lower part of the spider plot, you can see that's showing participants whose tumors have shrunk, and you can see how long that specifically lasted. And you can add different types of diamonds or circles or different colors to further describe what has occurred. You might see an individual who had progressive disease early on, but still the tumors that are being measured stayed down low for a longer period of time. So it just provides more granularity, if you will, more definition associated with the plot. So I'll go to the next slide. Now we're going to move into taletrectinib. So taletrectinib is a next generation potent and selective ROS1 inhibitor. I'm going to define those terms as we move forward, but we'll hold off on that in this individual slide. It was designed to improve efficacy and safety, so to improve anti-tumor activity and to be very safe, and really provide long-term disease control in individuals that are afflicted with ROS1 positive non-small cell lung cancer. It has the ability to effectively treat CNS metastases. I will show you that. It has the ability to overcome acquired resistance to first-generation ROS1 inhibitors. We'll see some of that data. And it also has a selectivity window of its activity versus ROS1 as compared to TRKB. TRKB is another protein kinase. Its inhibition in normal cells is thought to result in some of the side effects we see associated with some ROS1 inhibitors and also TRK1 inhibitors as well, these neurological side effects like dyschezia, paresthesia, and dizziness, and some of the ataxia type effects. So you can see the structure of the drug on the left. We'll talk through this in some of the coming slides. If you could go to the next one, Janet. So this is an extremely complex slide, so let me take you through it slowly. The data in the graph on the right and the table on the left is exactly the same. It's just shown in a different way. It's the same numbers. And what we're looking at here is IC50s. So you can see this boxed in red at the bottom here, and it says IC50 represents a half maximal inhibitory concentration. What this tells us is really how active a drug is, how potent it is. What is the lowest concentration that's needed to inhibit 50% of ROS1 is what we're really seeing here. This is a commonly used measure in drug discovery to assess the potency of our individual molecules. So in the table on the left, this is a reaction that's done with the specific enzyme, the ROS1 enzyme, in these individual drugs in a test tube. There are no cells involved here. There are no animals involved here. This is 100% the kinase in a test tube with the individual inhibitor. And you can see these numbers, like for instance, for taletrectinib for ROS1 0.07, for TRKB 1.47. This tells us that taletrectinib is more potent because that number's lower against ROS1 as compared to TRKB. And then if we look at the graph on the right, what we're seeing is that window. So what we've done for the furthest left blue bar is we've taken that TRKB number and divided it by the ROS1 number. So 1.47 divided by 0.07. And that gives us fold selectivity. So that tells us it's more selective for ROS1 versus TRKB, and there's about a 20-fold selectivity against those two targets. So I realize this is super complex, so let's go to the next slide, and I'm going to show you how I think about this and trying to distill it down. It's really a simple process of trying to thread the needle, and we're doing that with this selectivity window. If you look on Google, what does threading the needle really mean? It says to find harmony or strike a balance between conflicting forces, interests, etc. You can think of those conflicting forces as targeting ROS1, but at the same time, trying to stay away from TRKB. And by that, we can target the protein that is responsible for driving the cancer and stay away from the protein that could be driving the side effects. So that's what we're really trying to do here. And that's what other ROS1 inhibitors are trying to do as well. So I showed you some of the selectivity data. If we could go to the next slide. So we know that taletrectinib is selective for ROS1, specifically versus TRKB. Now we're looking at some of these potency assays, again at IC50s, that are done specifically in cells. So we're going up one layer. We're started with the enzyme, now we're going up to cell-based experiments, and you can see those here. So in this case, the drug is going to have to get inside the cell for it to be active. So in this table on the left, you can see in a specific model, don't worry about where it says BAF3CD74. That just tells us the specific model and the specific type of fusion. The type of fusion is a CD74. I would worry about where it says ROS1. And if you look at the taletrectinib number, in a ROS1 fusion, the IC50 is 0.8 nanomolar. That's a pretty potent number. Importantly, it's also active against other specific clinically relevant mutations that occur in ROS1 in individuals that have progressed on prior ROS1 inhibitors, one of those being crizotinib. And you can see some of those crizotinib mutations on the right. So for instance, L1951R is the second one down. The IC50, very similar to what we saw for native ROS1 at 4.2 nanomolar. I'm not showing you what exposure looks like in humans, meaning what the drug levels look like here. But when you have a drug that's pretty potent in the low nanomolar range, for instance, for G2032R, we're at 13.5 nanomolar. If you have good exposure in humans, meaning when you take the drug, what does the levels of that drug look like in blood that eventually is available to go to your cells, you can predict that you're probably going to be in a range where the drug is going to be active in an individual human. So far I've shown you we have a selective profile, and we also have a molecule that's potent against ROS1, and it's also potent against specific mutations that occur clinically in individuals with ROS1 mutant lung cancer. So if I could go to the next slide. This is looking at the taletrectinib's ability to penetrate the brain and also be active in specific models of brain cancer with ROS1 fusions. So the table on the left shows concentrations of taletrectinib, both on the, if you look on the side of the left, the deeper blue, total concentration in the brain is the first column, and then concentration in the plasma, which is really reflective of the blood. And what we've done here is we've looked at a ratio. A ratio of 1:1 is going to have equal concentrations in the blood and in the brain. So you're looking for something that's approaching 1 or perhaps higher than it. So you can see ratios of 0.4, 0.9, or even 3 as we go across time. So this suggests that taletrectinib can penetrate the blood-brain barrier and have suitable levels in the brain to potentially address the cancer in that compartment. The blood-brain barrier can be quite challenging to penetrate sometimes. Well, this doesn't, penetrating the brain means one thing, is it really active in the brain? So the study on the left is showing a study in rats. The study on the right is shown in mice, where tumor cells with ROS1 fusions are implanted directly into the brain, and then we're looking at survival curves in these mice that have been treated either with taletrectinib or repotrectinib. And you can see in both cases that each of the curves does a lot better than vehicle. Vehicle meaning they're not getting any drug. So we're seeing increased survival in a model of brain cancer in mice. We're seeing penetration into the brain compartment. We're seeing a molecule that, again, can cover acquired resistance mutations and one that is selective for TRKB. So all of this is just preclinical modeling. What does it really mean when the drug gets into humans? And that's what we're going to go through now. So if you could go to the next slide. So this is the, at a glance, the taletrectinib clinical development program. We have completed on the left 2 phase 1 studies, one that occurred in the US, one that occurred in Japan, and we have multiple ongoing phase 2 single arm studies, meaning there's not a control arm. Trust C203 is a trial that's moving forward in China. That is the study that I'm going to talk mostly to you about today. And the data in that trial is consistent what we saw in larger global populations in the US and Japan. So we're really going to focus on that data. And then Trust2 G208 is our global trial that is open and accruing now. So we'll talk about that. Also important to note that taletrectinib has achieved breakthrough therapy designation, or BTD, for individuals that have ROS1 TKI naive or crizotinib pretreated ROS1 positive non-small cell lung cancer, both in the US and in China in 2022. This is very notable for the company, very important for us. What this is, at least from an FDA perspective, it means that in illnesses that could be seen as life-threatening or serious, that the preliminary evidence, clinical evidence of activity is potentially better than what the standard of care is. And you have to obviously file for this, and it's been granted in these 2 individual countries. It also helps expedite the review process of moving towards an approval as you go forward. So I'll go to the next slide. I realize I'm running a little over on time. I hope that's okay. I'm going to just keep pressing forward. There's a few slides left, but I'll try and move a little bit quicker. This slide here details the design of Trust1. Again, this is the study that was conducted in China. These are in patients with metastatic or locally advanced ROS1 positive non-small cell lung cancer. I would not worry about where it says section 1. That was a safety lead-in that was done. We will focus mostly on section 2. There was a cohort or group of individuals that had TKI naive disease, meaning they had not received a prior TKI, but they could have had prior chemotherapy, and then individuals that had failed prior crizotinib exposure. So if we could go to the next slide. These are the baseline characteristics and demographics of the individuals that participated in Trust1. Important to note that some of these folks had prior anti-cancer therapy, obviously no TKI in those that had ROS1 TKI naive disease, but there was some chemotherapy. Also important to note that some of these individuals had brain metastases, and obviously higher in folks that had prior pretreatment with crizotinib at 31%. So if you could go to the next slide. Now we're finally getting into some of the endpoints associated with taletrectinib in this trial. In the TKI naive cohort or group of individuals, the overall response rate was about 93%, 92.5%. You can see that boxed in. Again, this is the percentage of individuals whose cancer shrunk, and there were by greater than 30% or had a complete disappearance would be reflected here as well. And there were 67 participants in this cohort. If we look at the figure in the bottom right, this is the waterfall plot that I showed you before, and you can see there's consistent tumor regression that occurs across the majority of patients in this cohort. We can also see in the spider plot, if you remember the one we showed earlier where the lines were kind of all over the place, you can see a lot more fluidity and a lot more consistency here, where there's many patients whose tumors are shrinking and they remain shrinking for a good period of time. You can see also that 30% shrinkage mark, that line is shown, and that 20% growth mark reflecting the barriers for progressive disease or a partial response. We haven't talked much about PFS. PFS is progression-free survival. It's a measure of the length of time from the start of therapy to when an individual progresses. We have conducted those analyses associated with the trial. If we look at this just from Trust1, we haven't followed the trial long enough to really understand what that number is. However, if we pull that data with our phase 1 patients, which are very similar populations, we see a PFS of 33.2 months. And that is publicly available information that was presented last year at ASCO. Okay, I'll go to the next slide. And then this is looking at the cohort of individuals that had received prior crizotinib. The response rate is also quite high here. Again, this is a measure of tumor shrinkage, ORR at 50.5%. You can see in the waterfall plot, the responses are not as deep, but they are still deep in a patient population that has received prior crizotinib. Also on the right in the spider plot, you don't see as much of the same consistency, but you still see a good group of individuals who are responding and having durable responses. Important to note, I showed you the acquired resistance preclinical information earlier on. You can see the clinical information here. We had 5 individuals in the trial that had G2032R mutations. 4 of 5 of those individuals responded. So that's shown in the red box, and then the yellowish diamonds on the waterfall plot and respectively on the spider plot shows how those patients fared on taletrectinib. So let's go to the next slide.
Can I ask a question here, Mike, before you go on?
Yeah.
So of the group of people who have been previously treated, do you know what their all the resistance mutations, were any of them off target and still responded?
So that's a great question. I don't know the answer to that right now, Janet, but we are going to be moving forward with analyzing that data further after we complete accrual associated with the trial to understand that. That also, what we'd want to look at is what the mutational profile looked like at baseline. That will be the easiest information to get from how from the translational analyses that occurred within the study. It's much harder, as you know, to get information after after individuals have progressed, getting biopsies or even getting blood to understand what the resistance mechanisms look like. So all I can tell you right now is we plan to analyze that and look into it further.
I see somebody unmuted. Was there another question? No.
This is Jerry. I just add to what Mike just said is that indeed there are the existing methods to measure those secondary mutation actually have their own limitations. So we try to based upon both tumor sample as well as liquid biopsy to get those information, and it does take time for labs to do those analysis. But what I can share is in this study, only about 40% of the patients have, as far as we know, based upon the information, have less than 40% have a secondary mutation based upon collectively of different analysis methods. The remaining over 60% of patients does not have any secondary mutation at all.
Well, thank you for that. That actually raises another question. So you mentioned tissue. Do the patients need to have available tumor tissue in order to join the trial?
Well, this is the China study. Initially, well, actually, for part of the trial, it was a requirement. But then we realized that because it's better to have the tissue to figure out what kind of secondary mutation the patient may have. But later on, we found out that for the crizotinib treatment failure patients, some of them, it's very challenging to get the biopsy. So we loosen the requirements. So for our global study right now, I believe, Lian, maybe you can chime in. We would not make it mandatory, but still for the trial perspective, if we can get the biopsy, it would be ideal for the trial.
Yeah, I think the Mike has a slide specifically talk about the tissue requirement. You will see. Yeah.
Yep, we'll talk more about that, about the global pivotal trial that's ongoing right now and where we are with respect to tissue requirements. But we've made it, I can tell you, we've made it as flexible as we think we possibly can.
Thank you.
Yeah.
Next
So let's
slide.
go ahead to the next slide. I told you that taletrectinib is a drug that we believe can impact disease that is spread to the brain. This is looking at individuals from the TRUST-I study that had measurable brain metastases, meaning they were greater than 1 centimeter in diameter. So there were 12 patients that had this form of a disease, and 11 of those 12 individuals responded, which correlates to an intracranial response rate of about 92%, 91.7. You can see the DCR rate, the disease control rate is 100%. This would include folks with stable disease, and shrinkage occurred in all of those patients, just greater than 30% in 11. So, and this is, to be important, to be clear, this is in individuals from both cohorts. Let's go to the next slide. Okay, we're at the safety. And then we'll talk about G208, our pivotal trial after this. So this shows the taletrectinib safety profile, which we believe is well tolerated across clinical trials. So this is pooled information from phase 1, from G2, from the TRUST trial that I just went through, as well as some other trials. You can see that the most predominant side effects tend to be elevation of liver enzymes and some GI-related side effects. The neurological side effects tend to be quite low. The rate of dizziness in this pooled population was about 18%. The majority of the adverse events that we see tend to be low grade, usually grade 1 or 2. They tend to be manageable and they tend to be reversible as well. Importantly, the dose reduction rate, meaning how many individuals actually decreased their dose from the standard dose, was quite low. It's about 14%. And those that discontinued because they had an adverse event was 5.3%, so quite low as well. Okay, so let's go to the next slide. I think there was a question. I didn't get a chance to read that. I don't know if you want to go through that really quickly, Janet.
Yes, there's a question. Patient is asking, is taletrectinib suitable for patients who have developed resistance to second-generation drugs like entrectinib or repotrectinib?
So that's a great question. From an enrollment perspective, I think this is a great segue into the next slide. If they had the right eligibility, they would be, these individuals could potentially join our trial. So let's go through the cohorts, and I think this will partially answer the question here. So there are 4 cohorts in the TRUST-II study. This is a global phase 2 study supporting registration of the drug. Cohort 1, the target enrollment is 53 participants. These are individuals with ROS1 positive non-small cell lung cancer. And again, this is TKI naive. So no prior TKI and up to 1 prior line of chemotherapy. Cohort 2 is ROS1 positive disease as well. These individuals can have 1 prior approved ROS1 TKI. That could be crizotinib or entrectinib, because these are the only 2 molecules that are currently approved, and up to 1 prior line of chemotherapy. Cohort 3 is a little more prior therapy. So again, ROS1 positive disease, up to 2 prior TKIs with ROS1 activity. So this doesn't mean they have to be approved. They just have to carry ROS1 activity, and up to 1 line of prior chemotherapy. And the final cohort is, or I'm sorry, I should say that says 2 or more TKIs. Apologies for that. And then the 4th cohort is ROS1 positive solid tumors, so beyond non-small cell lung cancer, or accommodating participants with non-small cell lung cancer who are ineligible for cohorts 1 to 3. So they can have up to 3 prior TKIs, and I believe up to 2 prior lines of chemotherapy, if I remember correctly. So I think to answer that really answers the question through the criteria associated with the trial. If these participants meet the eligibility criteria, we feel like we've tried to create space for all patients within this study.
So, Mike, I have another question here. I noticed that you allow locally advanced non-small cell lung cancer, which would be stage 3, I assume. So if that's true, do you also allow patients to have had prior immunotherapy?
We do. If Leanne or Heinrich, you can confirm this. I believe there's a 4-week washout that has to occur after prior IO. Is that correct?
Yes.
Thank you.
And
And
we
I
talked
see your,
about,
go ahead.
Okay.
I just saw your tissue requirements are also on there.
yeah, that's the one thing I really wanted to highlight here. So you see the enrolling countries. We are a global trial. You can see the enrolling countries below that occurs in Asia, Europe, Canada, and the US. And from a tissue perspective, in cohort 1, tissue is required, but is not required for enrollment. It can be retrospective tissue that's just provided for central confirmation of ROS1 status. Cohorts 2 and 4, we obviously would love the tissue. We recommend it, but it is not required. So if an individual had ROS1 positivity through a local test, they could obviously join the trial from that mechanism.
So to repeat on cohort 1, if these are people who've not previously been treated for ROS1, if they have a test that shows they are ROS1 positive, would you accept that test, or do you need to run a separate test?
So it's a CLIA validated assay, but I think it would be better for Leanne or Heinrich to provide that answer.
Yeah, so if the patient's ROS1 status can be, we accept the local assay result. We don't have to be the central confirmation.
Okay.
Is that helpful, Janet?
Yes, thank you. Also a question about enrolling countries. We have a large cohort of patients who are in Australia. Are you planning to expand to Australia anytime soon?
We are probably not going to move into Australia. I think we've made that decision, correct, Leanne?
Yes, at this moment, we are not, intent to enroll patients from Australia, but, we are opening the phase 3. So that's where we can go to the country in Australia.
You are planning a phase 3 study? What are you going?
We are planning. Yeah, we're planning phase 3 as well.
So if you do that, which drug are you thinking of comparing it to, if I may ask?
We're still debating. So basically still, the standard of care of the ROS1 TKI.
Okay.
Very likely, yeah.
It could be an either or, Janet. It could be crizotinib or entrectinib.
All right. Thank you.
But we are, very moving into deep discussions associated with what that trial will look like.
Thank you.
Beyond that, my final slide is just a summary, and this would just be redundant and reiterative information for you. But what I can tell you is we're extremely excited about this drug and the promise it can provide to individuals that have ROS1 positive non-small cell lung cancer. We think it's active in the first-line setting with or without chemotherapy. We think it's active after individuals have been pretreated with a first-generation ROS1 inhibitor. We think it's active and can treat brain metastases and can address specific acquired resistance mutations like G2032R. And most importantly, we think it can provide good quality of life, and we will be analyzing that in the G208 study, our global study, with a well-tolerated profile with low incidence of specific neurological adverse events. And we look forward to bringing the global data to FDA, supporting that BTD that we have. So I'll stop there. I'll take any questions that you have. I really appreciate everybody listening.
Thank you for that presentation, Mike. I have a couple questions. If there are others, post them in the chat or feel free to ask them. So you mentioned that you're going to be looking at quality of life. So do you have you established some patient-reported outcomes you're going to use to try and capture the quality of life?
Yeah, we are planning to use, my understanding is the standard quality of life instruments that are utilized in a lot of lung cancer trials. So the LC13, I think, is the lung cancer specific one, and the QLC, I can't remember the name of it, but it's the common ones that are utilized.
Yeah, we use 3 different questionnaires, and that's why specifically for UK, the E5EL, I forgot, and the LC13 and the QL30. That's very standard questionnaires. Yeah, we do.
Do any of those capture cognitive side effects?
For that, I don't know the detailed questionnaire. I may have to check back.
Yeah, I don't know, Janet, if there's a standard instrument that is good at capturing those that's utilized in the oncology community. You might know better than me. But I've heard that this is a challenge. Yeah.
Yeah.
Also, in terms of quality of life or cognitive, questionnaire. So while we are planning the phase 3, so we can do the CTCAE related questionnaire. So basically we can, among the more than 200 different items, I think there's must have the, cognitive outcome included. We can further explore that. Yeah.
Okay. So I know some other questions have come up in the group regarding enrollment. So one of them is that if you do not have the trial available in your country, is there anything as far as from your side as the sponsor of the trial that prevents people from enrolling in another country?
Not from a sponsor perspective.
I realize that sometimes the clinics or countries may have their own regulations relating to that.
Yeah, we don't have anything in the protocol that should limit that.
Yeah,
sorry, this is Heinrich. There is it's institution specific depending on where the out of country is, because it has implications on, of course, translations of informed consent and things like that. So, you get somebody that doesn't speak the local language, even if they're willing to travel. There are some other regulatory issues that might prevent them from participating if they're out of country.
So just to translate that to the rest of the group, ethics regulations usually require that a person go through the consent process in their native language, and a country may not have it set up to be able to support other languages, which would then make it difficult for them to enroll someone to the trial. That's what you're talking about, Heinrich, correct?
Correct. Thank you. Yeah.
Okay. Other questions? Well, that's a lot of information, and I really appreciate Mike and the AnHeart team coming on and sharing this information with us. I will be able to make the slides and the video available shortly after we get them set up. So thank you again for that, for allowing it.
Absolutely, Janet. Thanks so much for your time. Thanks everybody on the call for your time. And if any questions come up, please feed them through Janet, and you're welcome to bring them towards our team.
Well, thank you very much. We will be ending the recording, and then the meeting usually continues so that the patients can have a conversation. So I will ask the AnHeart team to sign off at this point.
Okay,
thanks.
thank you.
Take care, everybody.
Bye.
Thank you very much for the presentation. It was great.
Gives us lots of hope.
Thank
Yeah.
you. Take care.
Bye.
Bye-bye.